Hi class and welcome. I hope everyone's ready for our next lecture presentation. In this video, I will be discussing animal digestive physiology. The gastrointestinal or simply the GI system of an animal serves important functions. First is the feeding, that is, the delivery of food to the start of the GI tract. This involves ingestion and mechanical process of breaking down of food. Next is digestion. It is a process where the initial food that is eaten is broken down in both physically and chemically. Chemical digestion involves secretion of enzymes that degrades food molecules into its simple form. Next is absorption. It is where products of digestion are removed from the GI and transferred to all cells. In the cells is where the nutrients from the food are being absorbed. Last is the elimination or excretion, where foodstuffs which are eaten and cannot be digested are eliminated as waste products. A typical mammalian gastrointestinal system is consist of four basic regions. First is the reception and conduction region, which is composed of the oral cavity or the mouth and the esophagus, while the stomach serves as the storage region. Next region is the digestive and absorptive region, where both the stomach and small intestine are involved. And lastly, the water absorptive and excretive region, which is composed of the large intestine and the rectum. The receptive region of the GI system is the mouth, which is composed of the teeth involved in the gathering and mechanical breakdown of food. The mouth also secretes saliva by major salivary glands such as sublingual gland, submandibular gland, and parotid gland. Its primary function is for lubrication. It also contains amylase, an enzyme which begins the process of carbohydrate digestion in some mammals. Also part of the receptive region is the esophagus. It is a tube which connects the mouth to the beginning of the gastrointestinal system. It has no digestive function, but its contraction, known as peristalsis, is responsible for the movement of food particles to the storage region, which is the stomach. Organs of the storage region are enlargements of the anterior region of the GI system and are primarily involved in the storage of food with small amounts of digestion. In chicken, aside from the glandular stomach, the storage organ of their GI system is composed of the crop, which is a temporary storage of food, and the gizzard. The gizzard is a muscular sac involved in the breakdown of ingested food. It assumes contractile activity and mechanical breakdown of food particles, aided by the bird swallowing stones. These stones help to crush particles of food. This ensures that large food particles will not pass down the GI but are retained in the gizzard for further mechanical and reduction in size. In mammals, the principal job of the stomach is to store partially digested food known as the chyme. It also plays a role in protein digestion by secreting protease that break down protein molecules. The environment of most vertebrate stomachs is highly acidic. This high acidity is caused by HCl. HCl is secreted by parietal cells, which activates the protease enzyme such as pepsin. Pepsin are stored and secreted as inactive precursors called zymogens. In particular, for pepsin, its zymogen is known as pepsinogen, which is secreted by the chief cells that lines the stomach. In ruminants such as cattle, goat, and sheep, the stomach has become highly specialized. That is, it becomes multi-chambered, which is composed of the rumen, reticulum, omasum, and abomasum. This is for the digestion of cellulose. This is because ruminants are unable to digest the cellulose cell walls of the plant material they are ingesting. The reason for this is that the cellulose is a polymer of glucose joined together by beta-glycosidic links. The enzyme system for vertebrates is only capable of breaking down alpha-glycosidic links. In order to digest the cellulose in their diet, ruminants have evolved a symbiotic relationship with bacteria and protozoans in their rumen and reticulum. These microorganisms are able to break beta-glycosidic bonds. Microorganisms that resides in the rumen and reticulum digest the cellulose that the animal initially eats. The partially digested food is then regurgitated or bring the already swallowed food back to the mouth for further chewing before being swallowed for a second time. This time, the food bypasses the rumen and reticulum. This is achieved by the presence of a fold in the rumen wall and enters the omasum and abomasum, which can be considered to be a true stomach. The processes of digestion and absorption are undertaken in the midgut region of the gastrointestinal system. This is the region which constitutes the first part of the intestine. The digestive process which occur from this stage onwards are known as enzymatic, which is governed by enzymes. The aim is to break the major components of food such as carbohydrates, proteins, and lipids down into their constituent components which can then be absorbed and utilized by the animal. The enzymes responsible for the digestion of carbohydrates are known as carbohydrases. Their job is to break the glycosidic bond that links individual monosaccharides to form di, tri, and polysaccharides. In general, carbohydrases may be divided into polysaccharidases or oligosaccharidases. Polysaccharidases or amylases are enzymes which digest polysaccharides such as starch, glycogen, and cellulose. In vertebrates, it is secreted in the salivary secretions and is known as salivary amylase and also by the pancreas or known as the pancreatic amylase. The products of its digestion are glucose and maltose, an oligosaccharide which is digested to produce glucose. Oligosaccharidases are enzymes which break down di or trisaccharides. These enzymes, which include sucrase, maltase, trihalase, and lactase, are located in close proximity to the epithelial cells of the gastrointestinal system, which are responsible for carbohydrate absorption. The enzymes which are responsible for the breakdown of proteins are called proteases, which break the peptide bonds that link individual amino acids together in a peptide or protein molecule. Proteases may be divided into two broad types. The first of these are endopeptidases. These enzymes are responsible for breaking specific peptide bonds within a protein. There are a number of endoproteases which work at a low pH. They include pepsin, which is stored and secreted as pepsinogen. Other proteases have an alkaline optimum pH. Included in this group are trypsin and chymotrypsin, secreted as trypsinogen and chymotrypsinogen respectively. Trypsin is activated by another gastrointestinal hormone called enterokinase, which is secreted by the gastrointestinal mucosa. Trypsin breaks the peptide bond involving arginine and lysine residue, while chymotrypsin is activated by trypsin. It breaks peptide bonds involving aromatic amino acids. There are two known types of exopeptidases. These are aminopeptidases, which remove the N-terminal amino acid of a protein, and the carboxypeptidases, which remove the C-terminal amino acid. Lipid digestion is achieved by the activity of two groups of enzymes, the lipases and the esterases. Lipases break down triglycerides, a common group of dietary lipids. Major lipase activity comes from that secreted by the pancreas, while esterases are involved in the breakdown of structurally simpler lipids. The activity of this enzyme is aided by the presence of the bile. The bile will act as a detergent, preventing fat droplets in the GI system from aggregating together, thus increasing the surface area which is available for enzymatic attack. The products of digestion such as amino acids, monosaccharides, free fatty acids, and glycerol must be absorbed from the gastrointestinal tract for utilization by the animal. The process of absorption requires the movement of these molecules from the lumen of the GI tract across the membranes and into the cells. It occurs in the small intestine. So, in order to increase the surface area available for absorption, this region in the small intestine must be highly folded. Originating from these folds are many finger-like projections or microvilli into the lumen. This is a functional adaptation which serves to increase the surface area available for absorption. The absorption of many sugars, for example glucose and galactose, occurs by the process of secondary active transport. This transport is linked to that of sodium. This contrasts with the transport of other sugars such as fructose, which is transported by the process of facilitated diffusion. Having entered the cells that constitute the absorptive region of the gastrointestinal system, the monosaccharides must enter the circulatory system for it to be utilized by the animal. The transport of monosaccharides out of the cell that initially absorbed them occurs by either simple or facilitated diffusion. Amino acids are absorbed by secondary active transport in a process similar to that utilized in the absorption of monosaccharides. Different transporters exist for the following amino acids. So different amino acid transporters are involved for the transport of neutral amino acids, basic amino acids, acidic amino acids, and for amino acids proline and hydroxyproline. There is evidence that whole proteins may be absorbed by the process of endocytosis, where the cell engulfs the whole protein molecule and digests it. Absorption of fatty acids and monoglycerides occurs by simple diffusion. The products of lipid digestion aggregate together with bile salts to form micelles. The process is known as emulsion. So once absorbed, the free fatty acids and monoglycerides are converted back into triglycerides. The newly formed triglycerides are coated with a lipoprotein to form structures called chylomicrons. Chylomicrons then enter lacteals of the villi, which are a series of vessels that return excess tissue fluid to enter the systemic circulation via the lymphatic system. The final region of the gastrointestinal system is the hindgut. This comprises the large intestine. The remaining water that is absorbed in the large intestine is facilitated by the active transport of sodium out of the gut lumen. This is accompanied by the passive movement of chloride ions in order to maintain electrical neutrality. This generates an osmotic gradient between the lumen of the hindgut and the cells which line it, so water is absorbed by the process of osmosis. The hindgut is normally colonized by a variety of bacteria that produce nutrients such as vitamins as a consequence of their own metabolism, which are absorbed by the animal. The remaining semi-solid mass of undigested food is called feces, and this is expelled periodically into the environment. So that ends our discussion on digestive physiology. Again, if you have questions and in need of clarification, please feel free to contact me or post it in our Mole classroom. Thank you and have a good day. See you again in our next session.